Welcome back, everybody, to the second part of our look at the plant kingdom. Today we are going to be focusing on seeded plants, looking at gymnosperms and angiosperms. And if you've spent some time looking over the chapter, of course, you did the seed plant lab last week. So if you've been spending time with seeded plants, this should be relatively easy to understand. By virtue of the fact that you already have some background experience with the clades that are represented here. However, we will be digging significantly deeper into the gymnosperms and angiosperms than you might have done by just looking at the lab. So it is very important that you not lose sight of the fact that there is far more to the seeded plants than you may have already encountered by working through the lab. That being said, let's get started and take a look at seeded plants. So one of the major adaptations that allowed plants to ultimately come to dominate the landscape on dry land was their ability to reproduce in the absence of water. As we saw in the previous chapter, both the non-vascular plants and the seedless vascular plants had a reliance on water for reproductive purposes because they possessed flagellated sperm. You're going to see the retention of flagellated sperm in two of the clades of gymnosperms, but that has not, as it would turn out, limited the gymnosperms, and particularly these clades of gymnosperms, from still moving into areas that are drier and contain less water. That would be because not so much of the loss of the flagellated sperm, because clearly they've got flagellated sperm, but their gametophytes are still in close approximation to each other, so they need to rely on very little water for reproductive purposes. But the big advantage is that, as we will see, they have the ability to protect the gametophytes in a way that allows for the development of an embryo in the body of the parent, and it is in fact the embryos that are released into the environment, prepackaged in a structure that we refer to as a seed that developed from an ovule. And this is perhaps the single biggest advantage of seed plants. The fact that rather than releasing a haploid single-celled spore into the environment with all the risks that is posed by that, the haploid stage of the reproductive cycle of seeded plants takes place within the body, and particularly the reproductive structure of the body of the sporophyte. And the gametophytes are grossly reduced in size. They are very, very, very small compared to the gametophytes that we've encountered already. Even though the gametophytes are somewhat small, some might argue almost microscopic in some cases with the ferns. In this case, the gametophytes are very, very small and contained totally within the body of the sporophyte. They are contained within this structure called the ovule that I've already mentioned, and is the ovule that will ultimately develop into the seed that is released into the environment, taking with it a little embryonic plant that already contains embryonic leaves and an embryonic shoot and roots. And it's also packaged with nutritive material, usually containing some carbohydrates, some protein, and perhaps a little fat, or maybe a lot of fat, depending on the particular species that we're referring to, and in a very dehydrated condition, which preserves the ability of the embryonic plant to survive in the absence of appropriate conditions. In order to reanimate the embryo, you need to rehydrate the seed, but that seed can remain in stasis in this dried condition for years. It's been reported that people have been able to germinate seeds out of the Egyptian pyramids. I'm not entirely sure whether that's true or not. I've heard that that's been the case, but I've not seen anything in publication that would confirm that. But I can say that seeds remain viable, that is, they're capable of germination for hundreds of years. And that has been documented. So this is a very, very good way to release the next generation of plants into the environment. This all comes about because of the emergence of heterospory, the heterosporous condition that we started to talk about in the previous chapter. And the simple observation that you now have two separate sporangia that give rise to two separate types of spores, a megaspore and a microspore. And this takes place again within the body of the parental plant. The megaspore will eventually give rise to the egg, which once it is fertilized by the microspore, or more precisely, a haploid nucleus, which is referred to as the sperm nucleus, but we don't have a sperm in the traditional sense that we might think of as a sperm, contained within the pollen. So the microgametophyte is contained within the pollen, gives rise to the male gamete, in this case a sperm nucleus. When it pollinates the egg, that egg then gives rise, that fertilized egg then gives rise to the zygote, which divides into the little embryonic plant within the body of the ovule, and nutritive material is packaged within the ovule as the embryonic plant develops. Once it is ready to be released, it has finished its embryological development, then water is withdrawn, it enters a severely dehydrated state, it is reduced to around 15% water. That puts the embryo into stasis, and the seeds are then released, or what is now a seed, derived from the ovule, is released into the environment. If suitable conditions occur, and the seed can be rehydrated, a process called imbibition, then the embryonic plant breaks stasis and begins to grow again, develops into a seedling. The stem and leaves eventually emerge through the soil and begin photosynthesis and begin the support of the growing plant. And the remnants of the seed and the nutritive material known as endosperm within the seed then shrivels up, and the plant takes on a completely independent and self-sufficient or self-supporting lifestyle supported by the process of photosynthesis. So you can see within that brief description of the life cycle some adaptations that are clearly advantageous to seeded plants that allow them to become the dominant form of plant in the environment. In fact, when we get to the angiosperms, it's important to recognize that probably 80%, I think is the accepted number at the moment, about 80% of all plants are actually angiosperms. So even in comparison between gymnosperms and angiosperms, we are going to see some derived traits of angiosperms that give them even more of an advantage over gymnosperms that have allowed them to become the dominant form of plant in the environment. Let's move on and take a look at these adaptations in a little bit more detail. So here you can see the 5 derived traits that we are going to revisit on multiple occasions as we talk about seeded plants. So I've already introduced the concept of reduced gametophytes. The male gametophyte is actually the pollen grain. That's what we refer to as the male gametophyte. The female gametophyte is a large cell, single cell initially within the body of the ovule. It actually produces multiple haploid nuclei. You don't really see a fully multicellular structure. it's a little confusing because we expect the gametophyte to be a multicellular structure, but in both the gymnosperms and angiosperms, to talk about it as a truly multicellular structure is a little bit of a stretch when we compare it with the earlier clades that we've looked at, the non-vascular plants and the seedless vascular plants, where clearly the haploid generation is multicellular in every sense of the word. We also have to recognize the presence of heterospory, the heterosporous condition, giving rise to the microspores and the megaspores, the role of the ovule, the reproductive structure that will give rise eventually to the seed. Here you see the gymnosperm version. So the megasporangium gives rise to the megaspore. That will in turn give rise to the megagametophyte, which gives rise to the egg. But it all takes place within this structure called the megasporangium. And you can see that we don't identify multiple cells either within the megasporangium or within the megaspore. Pollen is the other side of the equation, the male reproductive structure representing the highly modified sperm. So within the pollen grain, which is the male gametophyte, you'll have multiple haploid nuclei, one of which will represent the sperm nucleus. In gymnosperms, it is not unusual for there to be multiple germinations of eggs on the basis of multiple haploid nuclei within the pollen grain. In angiosperms, as we'll see, there are usually two haploid nuclei within the pollen grain, one representing the sperm nucleus and the other representing a haploid nucleus that will be utilized, because we talk about the idea of a double fertilization in angiosperms. More to come when we touch on angiosperms. Then, of course, the ovule ultimately develops into a seed protected by a seed coat, which forms from this fleshy membrane that surrounds the ovule called the integument. When it dries out, it gives rise to the seed coat. Packaged inside the seed is the little embryonic plant with embryonic leaves, an embryonic shoot, and embryonic roots, and the nutritive material, otherwise known as endosperm. So these are the 5 derived traits that we need to pay attention to. And I'll spend some time when we talk about the 2 life cycles that you need to know to identify where we see these 5 derived traits emerging within the life cycles that you need to be aware of and need to know. So what's the advantage of a reduced gametophyte? Well, remember the gametophyte and gametophyte cells are haploid. And of course, hopefully you remember that means that they have a single copy of each of the chromosomes, and therefore a single copy of each of the genes on those chromosomes. And the problem for plants that have gametophytes in the open is that they are subject to UV radiation and therefore subject to mutation. So in closing, the gametophytes within the sporophytes protects against mutation and the other advantage, it also prevents drying out. gametophyte. And you might ask, why is that a big deal? Well, remember, the gametophyte is where the gametangia are going to be located that are going to give rise to the gametes. If the gametophyte dries out, that's not going to happen. No more reproduction. So being able to protect the gametophytes from drying out means that you now can produce the necessary gametes for reproductive purposes and maintain the continuity of the plants from one generation to the next without having to worry about the gametophytes drying out. So this would be another reason why those plants that either have a gametophyte dominant life cycle, the bryophytes, or have a independent gametophyte, the seedless vascular plants, are still found in very moist environments, because that is the only way to protect the gametophyte from completely drying out or from the dangers of drying out. Here now we have an opportunity to prevent the gametophyte from drying out by simply enclosing it within the body of the sporophyte. Reduce its size down to only a handful of cells rather than a large multicellular structure, and enclose it within a very waterproof coating that contains sporopollen within the body of the sporophyte. It now runs very little risk of drying out, and therefore the reproductive cycle can continue even in environments where there is reduced amounts of water or a lack of water completely. I mean, after all, we have to remember that deserts might appear to be lifeless, but they are not. Both plants and animals can live quite comfortably in deserts as long as they're adapted to it. And so the ability to retain the gametophytes as far as plants are concerned in all environments is a distinct advantage. And so particularly angiosperms, not so much gymnosperms, but as we'll see, even some gymnosperms have the ability to live in environments that otherwise might be extremely hostile to plants that require substantial amounts of water. So that's a real advantage of having a reduced gametophyte. And so just to remind you of the change in relationship that takes place during the evolution of the plant kingdom, which is something that's really important to bear in mind, is that in the most basal groups of the current plant kingdom, and therefore most likely amongst the ancestors of the plant kingdom and the early members of the plant kingdom that started the colonization process, the gametophyte is the dominant stage. And the sporophyte is completely supported by the gametophyte. It is grossly reduced and completely supported by the gametophyte and is transitory. When we get to the seedless vascular plants, now we have a separate and independent gametophyte and sporophyte generation. Bear in mind that when you look at the fern life cycle, the life cycle that you are responsible for there, it does appear that the sporophyte may have some dependence on the gametophyte, but that would be an incorrect interpretation. It only looks that way because the sporophyte, or let's start with the zygote, is contained within the archegonium initially, but as it grows and emerges as a seedling, it begins photosynthesis and can support itself as a consequence, and therefore no longer requires any attachment to the gametophyte or any support from the gametophyte. And the gametophyte dries out, shrivels up, and disappears, leaving the sporophyte as a free-living generation. So we've gone from a sporophyte completely dependent on the gametophyte and a gametophyte dominant life cycle to a life cycle that is dominated by the sporophyte but contains an independent living gametophyte. Then when we get to the seed plants, and this is common across all seed plants, whether gymnosperms or angiosperms, now we have a gametophyte that is grossly reduced in size, completely encapsulated within the body of the sporophyte, and completely dependent upon the sporophyte for support, for nutrition, for protection, everything. The gametophyte is completely dependent upon the sporophyte. So we've seen a total 180-degree turnaround in terms of the relationship between the gametophyte and the sporophyte. You start out with gametophyte dominance and dependence of the sporophyte on the gametophyte, to a sporophyte dominant life cycle, but an independent gametophyte stage. And then you go to a sporophyte dominant life cycle with a gametophyte that is reduced in size, contained within the body of the sporophyte and totally dependent upon the sporophyte. There's your 180-degree turnaround. That turnaround can only be finalized with the emergence of seeds. So this dependence of the gametophyte on the sporophyte is part of the evolution of seeds. And we'll see that as we talk about the evolution of seeded plants. So the other thing that seems to be required for the emergence of seeds, the evolution of seeds, is the emergence of the heterosporous condition. And I kind of laid that out for you in the previous chapter, but let me just remind you of why we make this statement that heterospory is apparently required for the evolution of seeds. And that's because in every situation where you have seeds present, you also have the heterosporous condition. There are no seeded plants that are homosporous, producing a single type of spore. There are in the fossil record evidence of heterosporous plants that don't produce seeds, which does not break the principle. The principle is that seeds require heterospory, which means you can't have a homosporous plant bearing seeds. And we don't see evidence of that in the extant members of the plant kingdom, nor do we see any evidence of that in the fossil record. All extant plants that carry seeds are heterosporous, and in the fossil record, where the heterosporous condition exists in the absence of seeds, they're still heterosporous. It does not destroy the principle that seeds require the presence of heterospory. And in fact, we would expect to find plants that are heterosporous and not producing seeds, because somewhere in between homospory and no seeds and heterospory and seeds, there must be a transition from the homosporous condition to the heterosporous condition, and then the emergence of seeds. So not surprisingly, even the fossil record supports the requirement of heterospory as a prerequisite for the evolution of seeds. Then within the life cycle, we now see as a precursor structure to the seed a structure called the ovule, which is derived from the, I won't say derived, but it contains the female side of the plant structure. It contains the megasporangium, which gives rise to the megaspore, which will in turn give rise to what is referred to loosely as the megagametophyte, which will produce the egg. This is contained in a membranous layer. In gymnosperms, it's a single layer. In angiosperms, it's at least two. Some angiosperms have 3 layers of material called integuments, or membranes called integuments, that enclose this structure. At the base of the ovule is a small opening called the micropyle. And it is through the micropyle that the male gametophyte, in the form of the pollen grain, enters into the ovule in order to transmit the sperm nucleus to the egg cell, so for the purpose of fertilization. Once the embryo has completed its development, then the ovule develops into the seed, and the whole ovule develops into the seed. So when you look at a seed, say you cut open an apple and you remove the seed out of the core of the apple, That brown layer over the top, that's the seed coat that was developed from the integuments that surrounded the ovule. If you carefully tease that off, what you then have underneath that is a very thin protective layer that contains sporopollenin derived from the fleshy part of the gametophyte that is packaged within the ovule. It dries out and collapses down to form a protective layer over the top of the endosperm. So when you tease an apple seed open, you're going to see a lot of white material. That's actually the endosperm. If you very carefully separate the two parts of the apple seed, you'll see a tiny little lump on the surface of one of the two halves. That's the embryonic plant. It may not look much like a plant. It's certainly not green, but that's only because the development of chlorophyll, or at least the last couple of biochemical steps to the production of chlorophyll, require light. So a plant will not actually green up until the leaves are exposed to light as the embryo and then seedling emerge from the soil and get exposed to sunlight. Then you'll get it greening up as the last couple of biochemical steps to the development or to the production of chlorophyll take place. Pollen, as the male gametophyte, is produced in the microsporangium, which is located, depending on where we are looking, in a structure known as a staminate cone in gymnosperms, or in the stamens on the end of the anthers, in the anthers on the ends of the stamens, which represents the male part of a flower. Now, all of these structures are actually sporophylls. So when we start thinking in terms of a cone, when we start thinking in terms of a flower, we're starting to think in terms of sporophylls. And so because we have megaspores and megasporangia and microspores and microsporangia, we also have two different types of sporophylls. We have megasporophylls that contain the megasporangium, and we have microsporophylls that contain the microsporangia. More of that to come, particularly when we look at angiosperms. So here you can see the structures. Here's the ovule, the megasporangium, the megaspore as a product of that. There's the spore wall containing sporopollenin, preventing this from the megaspore from drying out. Remember, sporopollenin is the hydrophobic polymer that is unique to the spore walls of land plants. It provides them with a protective layer that stops the spore from drying out. We learned that in the last chapter. The megasporangium material is what collapses to form a protective layer adjacent to that sporopollenin, the spore wall. The spore wall, you can see as it enlarges, the spore wall continues to surround the material that represents the developing gametophyte. Within that is the egg nucleus. Here's the pollen grain that's entered in through the micropyle. It's germinated and produced a structure called the pollen tube. At the same time, within the gametophyte, the two haploid nuclei that are going to act as the sperm nuclei are produced. One of the sperm nuclei will fertilize the egg. What happens to the other sperm nucleus or the other haploid nucleus? It will likely just degrade. It is unlikely that it will fertilize anything because there is usually one egg per ovule. So it just gets lost. It just degrades, disintegrates. As the nutritive material expands, the megagametophyte, I guess, we should refer to it as that, the female gametophyte. The whole thing expands and is completely surrounded by the integument. When the embryonic plant reaches its mature form and is ready to be released into the environment, and the food supply has been laid on and is adequate to support the seedling, then the seed coat, the integuments, dry out and form the seed coat. And this is then made available to be released from the environment. In the case of conifers, there is an extension of the seed coat that extends up vertically up in this direction. Let me use the pen to just draw it in. It extends up this way. And it has an airfoil shape, so that when the seed drops, it starts rotating much like a helicopter blade. And that slows down the descent of the seed so that any air currents can push it sideways away from the parental tree. It's kind of cool to watch. It's a shame that we're not in the lab doing our labs on campus, because I could show you the seed dropping. But if you walk around the neighborhood and look under conifers in spring, when the seeds start dropping, you may see them lying on the ground, and you'll see the little knob at one end, which is the actual seed with the food supply and the embryonic plant inside. Then you'll see what looks like a little membranous extension. It's actually fairly rigid. But if you pick that up and carefully drop it again, you'll see that it starts rotating very much like a helicopter blade. And you'll see that it descends to the ground much more slowly than if it didn't rotate. And that's an advantage in the case of conifers, for distribution of the seeds. So those are the 5 derived traits of seed plants. And that brings us to our first break point and our first concept check questions. So I will break the recording here and see you on the other side with the answers to these 2 concept check questions. Compare how the sperm of seedless plants reach the eggs with how the same process occurs in seeded plants. And what derived features of seed plants not present in seedless plants have led to the success of seeded plants on land? I'll see you on the other side of the break with the answers.